is Biomed Cafe menu. Cell cycle regulators will be presented today. We will talk about direct regulators, indirect regulators, and quiz game. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. Cell cycle regulators. Cell cycle regulators can be divided into two groups: direct regulators or core regulators and indirect regulators. Direct regulators can be divided into 5 types: cyclins, CDKS, MPF, and APCC. Indirect regulators can be divided into 2 types: P53 and RB. Cyclins. Cyclins are a group of proteins that together work to regulate different phases of the cell cycle as core regulators. Function of cyclins: activating the cyclin-dependent kinases and activating some other proteins. Most of the cyclin proteins act by binding themselves to the cyclin-dependent kinases or CDK, which form a complex. The complex is then responsible for the regulation of the cell cycle. The concentration of these cyclins usually remains low for the most part, but peaks dramatically if they are needed during the cycle. The activation of the cyclin proteins is stimulated by the binding of the growth factors to the receptor on the cell, which activate the transcription of the cyclin genes. Cyclin types. Cyclins can be divided into 4 types: G1 cyclins, G1S cyclins, S cyclins, and M cyclins. G1 cyclins. G1 cyclins expression cycle. Their concentrations increase gradually with no oscillation throughout the cell cycle. Function of G1 cyclins: they accelerate G1 transition and they coordinate cell growth. Cyclin D is G1 cyclin. G1S cyclins. G1S cyclins expression cycle. G1S cyclins rise in late G1 and fall in early S phase. Function of G1S cyclins: they induce the initial processes of DNA replication and promote cyclosome duplication. Cyclin E is G1S cyclin. S cyclins. S cyclins expression cycle. The levels of S cyclins remain high not only throughout S phase but through G2 and early mitosis, as well to promote early events in mitosis. S cyclins induce DNA replication. Cyclin A is a S cyclin. M cyclins. M cyclins expression cycle. M cyclin concentrations rise as the cell begins to enter mitosis, and the concentrations peak at metaphase. M cyclins induce assembly of mitotic spindles and induce alignment of sister chromatids along the spindles. Cyclin B is a M cyclin. Cyclin-dependent kinase or CDK. Cyclin-dependent kinases or CDKs. Cyclin-dependent kinase or CDK is a group of enzymes that work to regulate different processes in the cell cycle. CDK is a part of enzymes consisting of serine or threonine units. The activity of CDK is only observed after the binding of a cycling molecule followed by the phosphorylation. CDK is inactive. Then, binding of a cyclin activates it. In addition to needing a cyclin partner, CDK must also be phosphorylated on a particular site, serine or threonine, in order to be active. CDK is a kinase that phosphorylates or attaches phosphate groups to specific target proteins. The attached phosphate group acts like a switch, making some target protein more active, or making another target protein less active. Maturation-promoting factor or MPF. Maturation-promoting factor or MPF. Maturation-promoting factor or Anaphase-promoting factor or MPF is the cyclin CDK complex that regulates the M phase of a cell cycle. MPF subunits. MPF has two subunits: a kinase subunit or CDK1 and a regulatory subunit or cyclins A and B. MPF is the regulator of the G to M transition. MPF function. Number 1. It triggers the formation of mitotic spindle through microtubules instability. Number 2. It promotes chromosome condensation through phosphorylation of condensins and histone H1. Number 3. It leads to the nuclear lamina depolymerization and nuclear envelope breakdown through phosphorylation of nuclear lamina. And number 4. It causes to the fragmentation of the Golgi and the ER through phosphorylation of GM130. Anaphase-promoting complex cyclosome APCC. Anaphase-promoting complex cyclosome or APCC. Anaphase-promoting complex cyclosome APCC is a protein that regulates the M phase of the cell cycle by inhibiting the action of MPF and causes the destruction of cyclin molecules. This molecule is important during the transition of a cell from metaphase to anaphase of the M phase. It adds a small protein tag called ubiquitin or UB. The target molecules are either M cyclins or securin. APCC function. Allowing the new daughter cells to enter G1. Step 1. APCC attached a ubiquitin tag to M cyclins. Step 2. It sends M cyclins to the proteasome. Step 3. In the proteasome, M cyclins are degraded. Step 4. The destruction of M cyclins pushes the cell out of mitosis. And step 5. It allows the newly formed daughter cell to enter the G1 phase. Another function of APCC is triggering separation of sister chromatids during mitosis. Step 1. APCC adds a ubiquitin tag to securin. Step 2. It sends securin to the proteasome. Step 3. In the proteasome, the securin are degraded. Step 4. Separase enzyme releases. Step 5. Separase chops off the cohesin that holds sister chromatids together, allowing them to separate. And step 6. the separation of sister chromatids indicates anaphase. P53. P53. P53, also called TP53 or tumor protein, is a gene that encodes for the protein that regulates cell proliferation and also acts as a tumor suppressor. In eukaryotic organisms, it is important as it suppresses cancer. The concentration of P53 in a normal cell is quite low. However, it increases due to DNA damage or stress signals. P53 function. Number 1. Cell cycle arrest. In the case of DNA damage, the cell cycle arrests by P53. It stops the cell cycle at the G1 checkpoint by triggering production of CDK inhibitor or CKI proteins. The CKI proteins bind to CDK cyclin complexes and block their activity and buying time for DNA repair. Number 2. DNA repair. In the case of DNA damage, P53 activates DNA repair enzymes. Number 3. Apoptosis. If DNA damage is not fixable, P53 triggers apoptosis, so damaged DNA is not passed on. Retinoblastoma protein or RB. Retinoblastoma protein or RB. Retinoblastoma protein is a nuclear phosphorylation that helps in the cell cycle regulation while also acting as a tumor suppression protein. RB prevents excessive cell growth during the cell cycle progression. It acts as a negative regulator of the cell cycle as inhibiting the process. Key points of cell cycle regulators. Cell cycle is regulated by regulators. There are two groups of regulators: direct regulators or co-regulators and indirect regulators. Thank you for watching. Please subscribe to this channel, and if you like the content of this video, please like it. Now it's time to the quiz game. I've put the link to the quiz game in the description of this video. Download it for free and enjoy it. Until the next video. Bye.